All right, people. So we are live with some more biology. Back again with Ms. Akili Joseph from the Australia term. And she is in a very pleasant, beautiful mood this morning. We are going to do some of her favorite part in biology as well, plant biology. Over to you, Akili.
All right. So hi, good morning or good afternoon, everyone. So today we'll be doing a Unit 2 question. Last week we did a Unit 1 question, so I'll help all the Unit 2 people this week. So we're going to be going back, throw back to 2010, looking at the 2010 paper. So I know this is like an old question and we always kind of want to do like the 2015 to more recent. But this question is an excellent example of CXC taking what you know and asking it in a slightly different way. So it's an oldie, but it's a goodie, right?
Nice.
So this is the 2010 paper. This is question 2 of the paper 2. And as I would have explained before, is that you know that how the 6 questions in your paper 2 coincide with your modules. So if it's question 2 or question 5, it's going to be testing your module 2 knowledge. Whereas question 1 and 4 will test your module 1 knowledge, and questions 3 and 6 will test your module 3 knowledge, right? So we can put on our module 2 hats now, for want of a better word, right? So jumping straight into this question, we see that, as I always say, read the fine print, read all the text regarding the question, just to make sure you understand what they want you to understand. And in this question specifically, that's very important because you would see a very familiar image, but they're using it in a different way. So for some reason, this version, when you download it from the CXC website, the diagram actually didn't display. So they actually used the same diagram in a separate question in the 2011 paper. So that's where I'm going to pull the image from for you to use, right? So, but let's get the introduction. So figure 2 shows apparatus that was used to investigate the distribution of stomata on the upper and lower parts of a leaf. And then they gave you the diagram, which should look like this, right? As I said, this is the diagram from the 2011 paper, but it's this exact same diagram. Let's look at the caption here. The caption for the image is figure 2, apparatus for investigating stomata distribution in a leaf. However, they use the same diagram for a different experiment the following year, saying apparatus for investigating the effect of environmental factors on transpiration rate, right? So we know this setup as the potometer setup, and we are accustomed to using this setup for investigating the effects of environmental factors on the transpiration rate. But we are not so familiar with using the same setup for looking at stomata distribution. The reason why I love this question is that they're using something that you're familiar with to ask it in a different way. So with this, the first question where they ask you is to explain 4 precautions that should have been taken when setting up and conducting this experiment, right? So when setting this up, you have to give 4 precautions, but if you don't understand how you're using this apparatus in a different way, then you may not be able to answer the question appropriately, right? So just a brief overview, you know that this is a conical flask, this is filled with water. You have your piece of branch of a particular plant. Here, this is a rubber bomb, this is completely sealed, and you should really have no air in this system, right? And normally water will be lost through stomata. Transpiration is the loss of water through the stomata in your leaves, right? So water would be lost from your leaves, and then it would cause the liquid to move in this graduated capillary tube, and you would usually look at the distance it moved to see how fast transpiration is happening, and then we manipulate different factors, whether it's humidity, light, wind, to see what effect it has and how quickly this line will move, right? But that's how we are accustomed to using this setup. But what they're doing in this particular question is looking at stomata distribution. So this is where you had to kind of put on your thinking hat and be like, well, how would I use this setup to look at stomata distribution instead? What you do here is basically to look at stomata distribution, they want to look at it as in the number of stomata at the top of the leaf versus at the bottom, right? So basically I'm going to tell you what you needed to do, but this is something that you would have had to kind of like think about in the exam for yourself. In order to use this setup to investigate stomata distribution, you want to set up this exact apparatus twice, right? And if you want to look at the number of stomata, the more stomata you have is the faster transpiration will happen and the faster this liquid will move down the column this way, right? So on one setup, you would put something that would prevent water from escaping through the stomata at the top of the leaves. So then you would coat all the leaves in your bunch with Vaseline to the top. At the top of all the top surface, the upper surface of all the leaves, you coat with Vaseline so that water can't be lost through the stomata. So therefore, any water that is lost from stomata at the bottom of the leaf because that is unsealed. And then the separate or the second setup, you would put Vaseline at the bottom of all the leaves. So the stomata, only the stomata to the top of the leaves would allow for water loss, right? So knowing to set up the apparatus twice and to use Vaseline is basically your novel way of using the same setup for the particular experiment that they're testing in the question, right? So because you have to set it up twice, this is where your precautions will come in. So one precaution you can take is that you want to ensure that the plant species, the leafy shoot that you're using here, is that it's taken from the same plant or the same variety of plant. You can't have a piece of mango branch and a hibiscus and looking at upper and lower because the distribution of stomata will be different across species. Right. So one precaution is you're using the same variety, right? Even to refine that precaution even more, you might want to say, okay, well, you're using branches from the same plant, right? To even make it more similar between the two setups, right? And then the third thing you want to look at with the leafy shoot is that you also want to make sure that the two bunches that you have, that they relatively have the same number and size of leaves, so that, you know, you want to make the both setups as similar as possible. So the only variable that's going to be different between the two of them is that which surface, whether it's the upper surface or the lower surface, that is sealed. Okay? Then you have your general precautions that you can use for this setup that you're accustomed to for whatever reason you're using this apparatus for. So you want to make sure that there's no air in this setup of the apparatus. You want to make sure that the seal here is very tight so that air is not coming into the system. This is water you can have here. You might want to add a dye to the water so it's easier to see the liquid moving along the graduated capillary tube. Another one is that you will want to, when you set up both sets of apparatus, you want to give it a chance to settle. So let some transpiration happen, and then you squeeze down your syringe in order to move this meniscus back towards the end of the tube. So you're resetting the apparatus before you start to record measurements, right? So any one of those precautions that I have said there before, one specific to the experiment that they're asking us to apply it to, as well as precautions associated with the general setup of this apparatus, would work for answering the first part of the question here, where it says to explain 4 precautions that should be taken when setting up and conducting this experiment. So the beauty of this question is that it's very practical, right? So anything that you propose that makes sense, you can get marks for. They're supposed to look at, once it's a logical response, you should be able to earn marks for it, right? So they only ask for 4, but there is over 10 to 15 possible correct answers, as long as it helps the experiment to be conducted appropriately, right? The only caution I would give for this question here is that they said explain 4 precautions and not list 4 precautions. So I always advise students that at the back of your syllabus is a glossary, and that glossary usually has your examining words, whether they say discuss, explain, outline, compare, or differentiate. So what are they asking you to do when they use these words, right? So for instance, because they said explain and not just list, right? You will want to give like no paragraph, no essay, just like a one liner or a brief description as to whatever precaution that you outline, right? So if it is that you said that you want to ensure that both bunches have the same number and size of leaves, you're going to say ensure that both bunches have the same number and size of leaves to ensure that they have the same number of stomata on their upper and lower surfaces. So just providing a brief reason, so that is an explanation as opposed to just listing a precaution, right? Because sometimes when that keyword of explain versus list means that if you only list it, you might only get half the marks because then the other half is for your explanations. So you because you don't have access to the mark scheme before the exam, you kind of want to just be safe and leave no doubt with the examiner, right? So we do exactly what they ask us to do. So that will take care of explaining your 4 precautions for the setup.
Got it.
Great, right? So then part 2 of that question says, from which surface will the water loss most likely be greater, right? So again, I'm just bringing us back to the diagram here. And this question should be able to be answered just from a theoretical point of view and not necessarily practical, because it's a fact that the lower surface of leaves will always tend to have more stomata than the upper surface, right? And that's because the upper surface one has that waxy coat layer, protective layer to the top. And remember, sunlight is usually hitting the top of leaves. So with sunlight hitting the top of leaves, if we had more stomata to the top of the leaves, the plant would be losing water via water vapor too quickly, right? So just plants have adapted to just having most of their stomata on the lower surface of the leaves. So from a theoretical point of view, you could have answered that question. But again, if you think about it from a practical aspect, you would expect that this meniscus here to move faster on the setup that had Vaseline to the top of the leaves, leaving only the lower surface, the stomata to the lower surface exposed, right? So for part 2, from which surface will the water loss most likely be greater? It would be greater from the lower surface, right? Or the lower part of the leaf. So then they ask us for part 3, suggest an appropriate control for the experiment, right? So when you set up a control for the experiment, it's something that you can compare to ensure that the results that you observe or the conclusions that you make is because of the manipulation of the variable that you're changing. So remember, we have 2 setups, with one having Vaseline on the upper surface and the other having Vaseline on the lower surface. And the only difference between the 2 of them, right? But how are we to know that there isn't probably some other variable that we're not thinking about that is affecting how quickly the meniscus is moving on the apparatus, or basically affecting how quickly water is being lost? So this is where a control would benefit us in the experiment. So a control for this particular experiment would be, we have 2 possible controls we can set up here. One, you can set up the apparatus that has no Vaseline on either the top or the bottom surface to see basically what the maximum or the fastest rate of water loss would be. So then if it is that we have a third setup now with no Vaseline on the top or the bottom, we would expect transpiration rates to be highest in that apparatus. So then we can see the effect of coating either the top or the bottom surface on transpiration rate, right? And remember, the transpiration rate, or the amount of water being lost, the conclusion we're making here is that the more water being lost means that there is more stomata or more holes for the water vapor to move through. So one control would be to set up this apparatus, no Vaseline on the leaves, to know what is normal transpiration rate, or what is the normal amount of water to be lost from your setup. Another control that you can do is to set up the apparatus again, but put Vaseline on both the top and bottom surfaces, so that you're basically sealing off the entire plant. You're sealing all these stomata. So then you can compare what your baseline, if there was no stomata present.
One of the controls is to have what the plant open, and the next one is to seal up everything, lock
Yes.
up everything.
So one is to leave all the stomata open, and the other one is to basically close all the stomata. So how you would close it is by putting Vaseline over all surfaces, right? So again, so for 1 mark, you can say either set up the apparatus with no Vaseline on any of the surfaces, or you set up the apparatus with Vaseline on both the upper and lower surfaces, right? And that would give you your 1 mark. So then they ask us to identify 3 factors that affect the closing and opening of stomata and explain how these factors function. So yes, it's only 3 marks, but you have to put, so I like to structure my answers as 1, 2, 3. If they ask for 3 factors, I put 1, 2, and 3 and write my factors next to it, right? But it's not only about identifying the factors, but it's also about explaining how these factors function. But generally, this is a 3-mark question, so you don't need your whole life story, right? Once you give a 1-liner explanation for your factor, you'll be satisfied that you've provided enough to the examiner to warrant them awarding you that mark, right? So one factor can be sunlight. So the presence or absence of sunlight means whether the plants can photosynthesize or not, right? If the plant can't photosynthesize, it's not going to want to open its stomata to get the raw products it needs, because that's how carbon dioxide comes into the plant, right? So one factor could be the presence or absence of sunlight, and then you have to explain saying if sunlight is absent, the plant can't photosynthesize, and therefore all its stomata would remain closed. So that would affect the stomata being more closed than open. And the vice versa, or the converse of that is true, that if sunlight is present, then the plant can photosynthesize, and therefore it's going to want its stomata to be open, right? Another factor could be humidity, right? So the humidity is the amount of water vapor in the air surrounding the plant. So if it is that the air is very humid, meaning that there's a lot of water vapor in the atmosphere around the plant, then the plant will feel safe in opening its stomata, because the rate at which water is going to diffuse out of the stomatal pore will be much lower. So therefore, having high humidity is associated with your stomata being open, more open as opposed to being closed. Converse of that is also true. So if you have low humidity, that means the air around the plant is rather dry, then you would find that the water vapor that's in the plant will diffuse out of it at a faster rate. And if the plant is starting to lose water too quickly, it's going to protect itself by closing the stomata. So again, you just identify a factor and you give your explanation. Yeah, and then I'll suggest a 3rd factor just for completeness. A 3rd factor could be wind speed, right? So if it's very windy, it means that all the humid air from the water vapor that is slowly being lost from the plant around the plant, wind is moving that water vapor away. So therefore, more water vapor from the plant is going to continue to diffuse out, and then the plant will close its stomata as a protective measure. Whereas if you have low wind speed or no wind present, then it's going to allow the humidity around the plant to build up. The plant feels safe, and it will continue to leave its stomata open, because the rate of diffusion of water vapor through the stomatal pores is going to be lower, right? So again, this is another question that you can have multiple factors. You pick the 3 that you're most comfortable explaining, and you give a brief explanation after it in order to ensure that you get your full 3 marks for this question. Yeah? And lastly, for part C, they give you this image, right? So they say this image, figure 3, is an electron micrograph of a plant structure, and they want you to tell them what are the structures they labeled X and Y, right? So again, this is testing our theory, and you should be able to identify this image knowing that this is an example or a section of a phloem tube, right? So this is your sieve tube and its associated companion cell, right? So we know that the sieve tube usually lacks a lot of the major organelles. So the reason it has its companion cell is to help it with some of the functions it uses the organelles in your companion cell to carry out the functions that they're lacking in the sieve tube element, right? So X is your companion cell. That's the cell that's usually right next to your sieve tube element. And then Y would be your sieve tube element. So you get half mark for each. It just said identify, so you can identify it by just writing the two. Companion X, companion cell, Y, sieve tube, or you could even be more specific and say sieve tube element. And so yes, so they'll be coming up to the last 5 marks of the question. They said to discuss 2 structural differences between your companion cell and your sieve tube element, and describe how, in spite of these differences, they function as a unit. So 2 structural differences you will see is that one, in your sieve tube element, it lacks all these organelles. It doesn't have a nucleus, it doesn't have ribosomes, it doesn't have a cytoskeleton, it has very little cytoplasm. All this reason because it wants more space for the products from photosynthesis to be able to move through these tubes, to go where they need to be, to go to the cells that need it, or to go to storage, right? So your sieve tube element's not going to have all these organelles while the companion cell has it. So one structural difference is that the companion cell has the nucleus, whereas the sieve tube element does not. The companion cell has ribosomes, while the sieve tube element does not. So again, any one of those organelles that the sieve tube element is lacking, that the companion cell does, you can identify that as a structural difference, right? So that's it too. And then how they function as a unit is because one of them provides services that the other one cannot, right? So for instance, your companion cell has ribosomes, which is key in making proteins that need to be present in your sieve tube element, but they can't make for themselves because they don't have ribosomes. So they work as a unit because one is providing or helping the other one get all the resources and the components that it needs, right? So that is how you would answer and earn your 4 marks for this question. So again, we're looking at the keyword here. It says discuss, right? So when they ask you to discuss, normally I say don't give a life story. You might want to talk a little bit more, so add appropriate detail to your structural differences here, right? And you're seeing how they work together as a unit, right? So again, some of those organelles that the sieve tube element lacks that the companion cell has would be a nucleus, ribosomes, mitochondria. Like mitochondria is a keyword I love to use because we all very comfortable with mitochondria being the powerhouse of the cell. So if that one has mitochondria and this one doesn't, then it's helping, it supports with the need for energy. Lastly, they said for part 3, name one route by which substances move from the chloroplast in the leaves to the phloem, right? So from the chloroplast in leaves is where glucose is made, right? And then converted to sucrose and it's transported as sucrose, right? So we want to get that all the components that is made in the chloroplast, it needs to enter the phloem in order to go to other cells that need it, that don't photosynthesize, or to go to your storage organs, say for instance in your root tubers or whatever, right? So this question is a theory-based question. So when you would have covered your content, you should know that there are various pathways in which they can move through, and that would be your apoplastic pathway, your symplastic pathway, or through your palisade. Does cacti have xylem vessels? I believe they do. They do, but they're more efficient at water storage. So that's why they can survive in very arid or dry environments. But their water conserving mechanisms on 100, on professional level, on elite level. Right, Candy? So yes, so just naming one route, you want to go with either apoplastic or symplastic pathway, and basically your theory would really give you details on the difference between the two, right? So Yeah, you just name, again, they say name, so you don't have to explain it. You just put the term ecoplastic pathway or symplastic pathway, and you would be golden for your 1 mark here, and that is how we would earn our full 15 marks for this question, right? But again, the beauty of this question here is I really wanted you to appreciate the fact that they can take a setup that you should be very familiar with in one aspect and basically challenge you to apply it in a different situation. Okay, so yeah, that's part of the things that we do in classes at the Student Hub, is that we're right now in the KPI classes, we're doing past papers, and while we're doing one question, I'm usually asking, connecting questions. So how else can we use this apparatus? So, etc., so that you get more knowledge, so that if questions come like this, testing something but in a different way, you'll be prepared.